Hi, I am Dr. P.R. Padma, Professor in the Department of Biochemistry, Biotechnology and Bioinformatics at the Avinajlingam Institute for Home Science and Higher Education for Women, Coimbatore. You have now chosen to do the module on the applications of plant tissue culture in food industry, which is part of the Food Biotechnology paper. In the last module, we have learnt about the basic techniques of plant tissue culture, namely PTC, and how it has become a common technique that is being adopted for the propagation of useful plant species in vitro. In this module, we shall consider the major applications of plant tissue culture in food technology and food industry. This module is part of the Food Biotechnology paper under the subject of Food Science and Nutrition. It is presented to give the learner an insight into the biotechnological applications in food industry by way of discussing the applications of plant tissue culture in food industry. After completing this module, you will be able to know the various ways in which PTC can help in the production of various food components and understand the major applications of PTC in the food industry. As we have already learnt in the last module, plant tissue culture or PTC is a technique that uses an artificial medium to grow and maintain plant cells and tissue under controlled laboratory conditions and then preparing them for the field conditions where they can further grow and produce the yield. Apart from this, PTC also involves the maintenance of the plant tissue itself or the undifferentiated mass developed from the plant tissue in culture, which is called callus, and mass produce the desired product in the laboratory itself using bioreactors. In both these methods, PTC has influenced the food industry immensely, and the field of food biotechnology is a booming business worldwide. Some of the major applications of PTC in food industry are generally in combination with other techniques like genetic engineering and gene transfer techniques. However, some applications like the production of some flavoring agents and food colorants have been achieved using plant tissue culture per se. Food technology and food industry have benefited from plant tissue culture in various ways. The most important among the applications include the following: production of high-value food additives, increase in the productivity of food and food components, improvement in the quality of food grains, vegetables and fruits. All these applications involve the pre-harvest phase of the food materials. In the recent years, attention has also turned towards post-harvest applications of PTC in improving food materials. Let us first take up the common food additives that are produced by plant tissue culture. The advances in the technique of PTC has enabled the mass production of various classes of food additives like the flavoring agents, coloring agents, preservatives, sweeteners, bitter components, pungent components and so on. Food flavors which are produced from plant tissue cultures we will be considering first. Several plant-derived components have been used in enhancing the flavor of food preparations. Many such agents are added as additives during the preparation of food. With the advances in tissue culture techniques, many flavoring agents have been mass-produced in culture. The major advantage of such a production is that these agents are from natural sources, unlike the synthetic flavoring agents, and they overcome the agroclimatic limitations and can be produced throughout the year. The major flavoring agents that are produced using tissue culture techniques include vanillin, garlic, onion, basmati, citrus flavor, cocoa flavor, capsicum, celery, chamomile, coriandrum, grape, mentha, orange, peppermint, perilla, rose and saffron. Apart from these complex flavoring mixtures, single compounds responsible for specific flavors are also being mass produced in culture. Some examples of these include 2-isobutyl thiazole, which gives a tomato flavor, methyl ethyl cinnamates that produce strawberry flavor, methyl anthranilate, which gives grape flavor, benzaldehyde that gives a cherry flavor, menthol that gives mint flavor, and saffronol that gives saffron flavor. Apart from these, whole lemon vesicles have been produced and maintained in culture, which produces the flavor components of citrus fruits, especially that of lemon. Coming to the food colorants from tissue cultured plants, most of the synthetic colorants that are marketed are obtained from petroleum byproducts and therefore are not considered safe for consumption. Hence, the search for natural sources that could continuously provide the color components that can be used in food preparations has been a quest among the scientists. The development of plant tissue culture techniques provides a viable alternative which has been successfully used in the production of food colorants. Some of the popular food colorant components produced from plants propagated in culture include the following. Anthocyanin pigments of various colors have been prepared from cultured plants. The most widely produced pigments include pink colored pigment rosinidin from Catharanthus roseus, a red anthocyanin pigment from Daucus carota, a red pigment from Euphorbia milli or crown of thorns, yellow colorant pigment from Aplopappus gracilis, red anthocyanin pigments called chiconin from Lithospermum thesis, and purple anthocyanin pigments from Vitis vinifera or grapes. Apart from anthocyanin pigments, iridoid pigments like loganin are also found in high amounts in Catharanthus roseus. These are also used in the food industry as colorants. Carotenoid pigments of various colors are produced using cultured Prunus persica. This class of pigments are isolated directly from the fruits of plums and peaches. However, the production level is augmented using tissue culture as well. Another major coloring as well as flavoring agent is saffron. This is prepared from the stigma of Crocus sativus plants. Scientists have developed stigma-like structures from this plant in culture that produces the flavor components in high amounts. This technique has made the production of saffron cost-effective. Additionally, it makes it possible to mass produce these flavor components, that is crocin and crocetins, from Crocus sativus, which are the active compounds responsible for the saffron flavor and color. The next food additives to be considered are the natural sweeteners from tissue cultured plants. Non-nutritive sweeteners are in high demand as the incidence of diabetes is on the rise, as well as these are seen as low calorie alternatives to sugar in fitness regimes. Using plant tissue culture, scientists have devised methods to mass produce non-nutritive sweeteners from natural plant sources. These include a combination of flavone glycosides from orange peel, stevioside from Stevia rebardiana, thaumatin from Thaumatococcus danilou, 8,9-epoxy perilartin from Perilarkin kinesis, and hydrofluorine diterpenoids from pine tree rosin. Since these have the advantage of being extracted from natural tissue that has been mass propagated in vitro, they are preferred over the synthetic artificial sweeteners. Similarly, the pungent component of chili called capsaicin, which is widely used as a food additive, is being produced by plant tissue culture of Capsicum annum. This has an advantage of being produced in very pure form, which is highly desirable as a method that is preferable for meeting the market demand for capsaicin. Capsaicin also has some medicinal uses apart from being a food additive. Now, we shall consider the essential oils from tissue cultured plants. Essential oils are used in several food preparations, especially the exotic preparations and the continental dishes. Tissue culture techniques are used to produce the plant parts from which the essential oils can be extracted. For this, adventitious shoot cultures are very widely used. The commonly produced oils in this method are mint oil, chamomile oil, jasmine oil, aniseed oil and geranium oil. Apart from these food additives, some components that are used as food preservatives have also been produced from tissue cultured plants. The preservation of food produce is a big challenge that the food industry has to deal with constantly. Several types of damages occur to food produce as well as the processed foods. One such damage is oxidative damage. Many antioxidant compounds are being tried to retard or to prevent such damage. One such component is rosmarinic acid. Rosmarinic acid is a phenolic antioxidant widely used in food preservation. It is mass produced using suspension cultures of Coleus blumei and Salvia officinalis. In both the plant cultures, rosmarinic acid content is very high, amounting to 21% and 36% of the dry weight respectively. This also provides a viable alternative to meeting the demand for food preservation. Considering the other food components from tissue cultured plants, protoplast fusion technique of plant tissue culture has been used to achieve certain desired traits in the plants. Salt tolerance was induced in rice plants by protoplast fusion of rice and ditch reed. In vitro protoplast fusion has helped in overcoming the sexual incompatibility barriers in horticultural plants. New hybrids with increased fruit yield and better disease resistance have been produced in culture by protoplast fusion. Good hybrid plants have been developed by fusing the protoplast from citrus plants with other related citrinase species. Another plant tissue culture technique called somatic hybridization is also used for developing better hybrids of food crops. This technique has been used to develop intergeneric hybrid varieties of plants of Brassicaceae family. Similarly, somatic hybridization has been used to overcome the problem of decreased regeneration due to loss of chromosomes in wheat plants. This was achieved by the production of somatic hybrids using two types of wheat protoplast as fusion recipients and the protoplast of Heinaldi villosa as the fusion donor, which also acts as an important gene source for wheat improvement. Apart from these products produced in culture, other applications include those of genetically transformed plants in the food technology. Genetic transformation of cultured plant cells followed by the propagation of such transformed cells results in the production of transgenic plants. Transgenesis has been successfully used in achieving several desired effects that have benefited the food industry immensely. These applications can be discussed under the following major heads. Number 1, resistance to physical and biological stress conditions. Number 2, improvement of nutrient content or nutritional quality of the food crops. And number 3, increasing the keeping quality of the food produce. Let us consider them one by one, starting with the resistance to physical and biological stress conditions. The physical stresses that affect the yield of crop plants are called abiotic stresses. These include pest or insect attack, weed interference, viral diseases, and other stress conditions like drought and salinity. Transgenesis has helped in overcoming all these stress conditions and improving crop yield. The attack of insect pests on crop plants has been counteracted by transferring the Bacillus thuringiensis or Bt gene from the bacterium into the crop plants. When the insects feed on such plants, the gene product interferes with the gut absorption of nutrients in the insects, thereby killing them. Thus, the crop plants are not destroyed by the insect attack. Using this method, Bt corn, Bt potato, and Bt soybean have been produced, which are the most popular crop plants that have benefited from this technology. Other such transgenic plants are also being developed. Weeds are undesirable plants that grow in between the crop plants and compete with them for the nutrients in the soil and also the water used for irrigation. Thus, the weeds affect the growth and yield of the crops. Weeds can be treated with herbicides which will effectively kill them. However, herbicides do not differentiate between weeds and crop plants. Therefore, when a herbicide is sprayed in the field, the crop plants are also killed, resulting in yield loss. To overcome this, genes of herbicide resistance have been transferred into crop plants by transgenesis of cells from crop plants which are maintained in culture. The transformed cells are then developed into plants and transferred back into the field. When this field is sprayed with the herbicide, the crop plants are saved because they have the herbicide resistance, and the weeds die. Roundup Ready soybeans, alfalfa, corn, spring canola, winter canola, and sugar beets have been produced successfully by this strategy. Another approach is to provide resistance to viral pathogens by transgenesis. Viral attacks cause a biotic stress on crop plant. This not only decreases the yield but also leaves toxic residues in the crop produce. Transgenic approaches have introduced the genes conferring virus resistance into potato, papaya, zucchini, and sweet peppers, which has resulted in improved yield and quality of the produce from these plants. Next, we shall take up resisting abiotic stress conditions like drought and salinity. Drought or non-availability of enough water is a serious condition that affects the yield of crops. Similarly, increased salt content in soil and the irrigation water is also a serious problem. To overcome these, transgenic plants have been developed by transferring the genes that confer drought and or saline resistance to the plants. Drought resistant canola or rapeseed plants, drought tolerant maize and soybean, drought guard maize and wheat, as well as drought resistant rice have been successfully produced by transgenic approaches. Similarly, salt resistant transgenic corn, maize, wheat and rice have also been produced and cleared for field production. Now, let us consider the improvement of nutrient content or nutritional quality of the food crops. Golden rice is a phenomenal success story in improving the nutritional quality of grains. In the production of golden rice, three major genes involved in the synthesis of beta-carotene, which is the precursor of vitamin A, are transferred into rice. The production of beta-carotene gives the rice a golden yellow color and hence the name golden rice. This is expected to eradicate the deficiency of vitamin A in the developing countries. Similarly, rice with high iron content, canola with improved oil profile, corn and banana with multiple vitamin enrichment, and cassava with lower cyanogen glycosides and higher protein and nutrient content have been developed using transgenesis. In several countries, the efforts in agriculture have resulted in good yield of crop products and grains. However, huge post-harvest losses are encountered due to poor keeping quality of the crops. The shelf life of the perishable food components like vegetables, fruits and grains is a vital contributing factor in the final yield of the crop. Thus, efforts from various angles are concentrating on increasing the shelf life or keeping quality of the food components. One such angle is the transgenic modification of the crop plants. Flavor saver tomato was one of the first successful efforts in this front, wherein genetic modifications to block the enzymes involved in the ripening of tomato fruits were introduced. This caused a significant delay in the ripening process, thereby increasing the shelf life of tomato. This also led to significant improvement in the loss incurred during the transport of the fruits over long distances. Similarly, transgenic apples with a reduced expression of polyphenol oxidase, resulting in a reduction in the enzymatic browning when the apples are cut, has improved the keeping quality of the apples. Additionally, a variety of potato called the Amphora potato has been developed with a transgenic modification that decreases bruising during harvest. Apart from these regular uses of transgenic plants, genetic modifications have also been carried out to incorporate therapeutic applications to crop plants. This has been achieved by expressing genes coding for therapeutic proteins in the crops. The major applications in this effort include the following. Plants have been developed to produce drugs against Gaucher's disease, which was approved by the Food and Drug Administration in 2012. Tobacco plants and bananas have been modified to produce therapeutic antibodies, which are called plantibodies because they are synthesized by the plant. Maize plants have been genetically engineered to increase the conversion of its starch to component sugars for use in the production of ethanol as a fuel. Industrially valuable varieties of starch have been produced by genetically modified potatoes. Soybean oils have been modified to increase the oil types used in the detergent industry. To summarize, tissue culture offers several advantages over the conventional practices in the propagation of plants. The mass propagation of plants is made possible by growing them in tissue culture. Controlled growth of these plants enables the selection of plants with desirable traits. Furthermore, using somaclonal variation, it is possible to induce the desirable traits in the plants propagated in culture. PTC also helps in overcoming the seasonal variations in the production of plant-based food products, enabling round-the-year supply of tissue culture propagated material. Thus, in this module, the various applications of plant cells maintained and propagated in culture in the field of food technology have been considered. At present, the major bottleneck is the cost effectiveness of the technique, which is not entirely economical. However, the efforts of the governments and the scientists is moving in the right direction and will result in major breakthroughs in the future. With that, this is Dr. Padma signing off, wishing you all a wonderful learning experience. Thank you.